Imagine a world where inherited diseases are no longer a life sentence. Picture crops that resist devastating blights, or therapies that precisely target and eliminate cancer cells. This isn't science fiction. It's the promise of CRISPR, a revolutionary technology that allows us to edit DNA with unprecedented precision. This powerful tool is transforming medicine, agriculture, and our fundamental understanding of life itself. It's a genetic scalpel, capable of cutting and pasting the very blueprint of living organisms. But what exactly is CRISPR, and how does it work its molecular magic? CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. This complex name describes unique DNA sequences found in the genomes of bacteria and archaea. Scientists initially observed these repeating patterns without fully understanding their purpose. It turns out these sequences are part of an ancient immune system. Bacteria use CRISPR to defend themselves against invading viruses. When a virus attacks, the bacterium captures a small piece of the viral DNA and inserts it into its own CRISPR array. This stored viral DNA acts like a genetic mugshot. If the same virus attacks again, the bacterium can recognize it. This recognition triggers a powerful defense mechanism, leading to the destruction of the viral invader. The key player in this bacterial defense system is an enzyme called Cas9, or CRISPR-associated protein 9. Cas9 acts like molecular scissors, capable of cutting DNA. But it doesn't cut randomly. It's guided by a special RNA molecule. This guide RNA is engineered to match a specific DNA sequence that researchers want to target. Think of it as a GPS system for the Cas9 enzyme. The guide RNA leads Cas9 directly to the desired location on the DNA strand. Once Cas9 arrives at the target site, it precisely cuts both strands of the DNA. This cut creates a break in the genetic code. The cell then attempts to repair this break, and this is where the magic of gene editing truly happens. Scientists can exploit the cell's natural repair mechanisms. They can either disable a gene by introducing errors during the repair process, or they can insert a new, desired piece of DNA. This new DNA acts as a template, guiding the cell to incorporate the desired genetic change. Before CRISPR, gene editing was a laborious, expensive, and often imprecise process. Older techniques were like trying to fix a single typo in a massive book using a sledgehammer. CRISPR, by contrast, is like a highly accurate word processor. Its precision is unparalleled. Researchers can target specific genes with remarkable accuracy, minimizing off-target effects. This specificity is crucial for therapeutic applications, where even small errors can have significant consequences. CRISPR is also incredibly efficient and fast. What once took months or years can now be achieved in weeks. This speed accelerates research and development, bringing potential therapies closer to reality much faster. Furthermore, CRISPR is relatively inexpensive and easy to use. This accessibility has democratized gene editing, allowing more labs around the world to conduct groundbreaking research. It has truly opened the floodgates for genetic innovation. The potential of CRISPR in medicine is immense, offering hope for countless genetic diseases. One of the most promising areas is the treatment of inherited disorders caused by single gene mutations. Consider sickle cell disease, a painful and debilitating blood disorder. CRISPR is being explored to correct the faulty gene responsible for abnormal hemoglobin production. Early clinical trials show promising results, with patients experiencing significant improvements. Cystic fibrosis, another devastating genetic condition affecting the lungs and digestive system, is also a target. Researchers are working on delivering CRISPR components to lung cells to correct the underlying genetic defect. This could revolutionize treatment for millions. Beyond single gene disorders, CRISPR holds promise for complex diseases like cancer. Scientists are engineering immune cells, known as T cells, to better recognize and destroy cancer cells. This approach, called CAR T-cell therapy, is being enhanced by CRISPR to improve its effectiveness and safety. CRISPR is also being investigated for its ability to combat viral infections. Researchers are exploring ways to use CRISPR to directly target and disable the genetic material of viruses like HIV, potentially offering a cure for chronic infections. CRISPR's impact extends far beyond human health. In agriculture, it's revolutionizing crop improvement. Scientists are using CRISPR to develop plants that are more resistant to pests, diseases, and harsh environmental conditions like drought. Imagine wheat varieties that are naturally resistant to rust fungus or rice that can thrive in saline soils. CRISPR can also enhance nutritional content, creating crops with higher vitamin levels or improved protein profiles, addressing global food security challenges. For example, CRISPR has been used to create non-browning mushrooms, reducing food waste. It's also being applied to improve livestock, developing animals that are more resistant to diseases or produce more efficiently. In basic scientific research, CRISPR is an indispensable tool. It allows scientists to precisely modify genes in model organisms like fruit flies, zebrafish, and mice. This helps them understand gene function and disease mechanisms, accelerating drug discovery and development. By turning genes on or off, or by introducing specific mutations, researchers can mimic human diseases in animal models. This provides invaluable insights into how diseases progress and how potential therapies might work. Despite its incredible potential, CRISPR raises significant ethical questions. The ability to alter the human germline, changes that would be passed down to future generations, is a particularly sensitive area. Concerns include the potential for unintended consequences, the concept of designer babies, and issues of equitable access. Who decides which traits are desirable? Will this technology only be available to the wealthy, exacerbating health disparities? There are also technical challenges. While CRISPR is precise, off-target edits can still occur, leading to unwanted genetic changes. Delivering CRISPR components to the correct cells and tissues in the body remains a complex hurdle for many therapeutic applications. Ensuring the safety and efficacy of CRISPR-based therapies requires rigorous testing and careful regulation. The scientific community is actively engaged in discussions and developing guidelines to ensure responsible and ethical use of this powerful technology. The field of CRISPR is evolving at an astonishing pace. New CRISPR systems are being discovered, offering even greater precision and versatility. Base editing, for instance, allows for single-letter changes in DNA without cutting the double helix. Prime editing offers even more sophisticated search and replace capabilities. These advancements promise to overcome some of the current limitations and expand the range of treatable conditions. The future holds the potential for personalized gene therapies, tailored to an individual's unique genetic makeup. CRISPR is not just a scientific tool, it's a paradigm shift. It represents a new era in our ability to understand and manipulate life's fundamental code. As we continue to unlock its full potential, responsible innovation and thoughtful ethical consideration will be paramount. The journey of rewriting life's genetic code has only just begun. CRISPR offers a profound opportunity to alleviate suffering, enhance human well-being, and reshape our world for the better. It is a testament to human ingenuity and the endless possibilities of scientific discovery.